Hi, I am Dr. Arsalan and today we are going to solve the MCQs on genetics. The first question is, pea plant has 14 chromosomes. Here the number of chromosomes of pea plants are given, that pea plant has 14 chromosomes. How many linkage groups are there? The genes present on the homologous chromosomes which tend to be inherited at the same time are called linked genes, and these form linkage groups. And it's a thumb rule that the number of linkage groups is equal to the number of homologous pair of chromosomes present in an individual. Here the pea plant has 14 chromosomes, it means that it has 7 pairs of the homologous chromosomes. So the linkage group will also be 7. So the option B is correct, that there are 7 linkage groups in case of the pea plants. Next question is, organism with two different allelomorphs is, given options are homozygous for that trait, heterozygous for that trait, both or none. So allelomorph means that having the alleles. Allelo refer here to the allele, the alternate form of gene, and morph means the morphology or form. So the individual has different allelomorphs or different pairs of alleles. For example, the black and white, these both kind of alleles are present in this gene. So capital B and small b, such individual is called heterozygous individual because of having different forms or different alleles. So the option B is correct for there, that the individual will be heterozygous for that trait. Next question is, what happens when two genes in dihybrid cross are situated on the same chromosome? Given options are, proportion of parental gene is higher as compared to the non-parental type, recombinants, proportion of non-parental is higher, non-parental means recombinant type, are new forms of the alleles present in the population, both have equal proportion, or none of these. First of all, we should know about the dihybrid cross, the cross between two individuals which differ by two traits. For example, in case of pea plant, if the parent strain differ by two traits, for example, the shape of seed and color of seed, so this will constitute a dihybrid cross. In this case, there is a dihybrid cross and the two genes, these are located on the same chromosome. So it's obvious here that the proportion of parental type will be higher as compared to recombinant or non-parental types. The option A is correct. Next question is, Mendel failed to identify the gene linkage because of, given options are, independent assortment, crossing over, gene mutation, or all of these. It is pertinent to mention here that the Mendel laws were insufficient on the concept of gene linkage, and the Mendel did not explain the gene linkage in the homologous pair of chromosomes, how the linked genes are transmitted from parents to the offsprings. Rather, he was with the opinion that the genes are never linked and these are assorted independently. These are separated and arranged independent of each other and are distributed equally into the offsprings. Therefore, he presented the law of independent assortment. So here option A is correct, the independent assortment. Repeat the question. Mendel failed to identify the gene linkage because of law of independent assortment. Next is a historic question. Queen Victoria passed hemophilia genes in her family tree. Hemophilia is a genetic disease, and if we further explain it in the language of genetics, the hemophilia is caused by X-linked recessive gene. For example, this is X chromosome, then the hemophilia is a recessive gene, and the female has 2 X chromosomes. If she has 1 X chromosome affected with the hemophilia gene, then she will serve as carrier, while in case of males, if X is affected with the hemophilia gene, then the male will be affected by the hemophilia disease. And in case of the female, when both of the X chromosomes are carrying the H gene or hemophilia gene, in this case the female will be affected. Affected female, this is the carrier female, and this is the affected male. So it's a predominant male genetic disorder which most often affect the males. In case of hemophilia, the blood fails to clot, and such individuals or patients are called bleeders because minor cuts in the hemophilic patients can lead to death and may prove fatal. Now we'll explain the question. Queen Victoria passed hemophilia genes in her family tree. So it's an historic statement in biology that the family tree of the Queen Victoria was affected with the hemophilia. Her genes in England, Spain, Germany and Russia, these are the 4 countries are extensions of family tree of Queen Victoria. Affected with hemophilia, the gene was passed from the Queen Victoria. She herself was dash of hemophilia. Given options are victim, producer, carrier or immune. So the option C is correct here, that she was the carrier of hemophilia gene and her chromosomal condition was just like this, that the one of the X chromosome of her were affected. Next question is, the term gene linkage was coined by dash. Given options are G.J. Mendel, Hugo de Vries, W.S. Sutton or T.H. Morgan. The term gene linkage was coined by T.H. Morgan and the phenomena was also explained by him. Next question is, in 1909, a dash-eyed male fruit fly, the Drosophila melanogaster. The fruit fly is called Drosophila melanogaster. It's a biological name, so I have underlined it. So fruit fly appeared in the bottle culture of Thomas Hunt Morgan. Thomas Hunt Morgan is T.H. Morgan, which led him to the phenomena of gene linkage. Which kind of Drosophila melanogaster or fruit fly male appeared in the bottle culture of the T.H. Morgan experiment? So given options are red, white, wild type, or homozygous recessive. So the option B is correct here, that a white-eyed male fruit fly appeared in the bottle culture of the T.H. Morgan experiment, in which the cross was made between the fruit flies without white-eyed colored. Recessive genes emerged here which rendered him to the phenomena of gene linkage. In a test cross between A and B, both served as linked genes. So the gene A as well as the B were the linked genes, were transmitted to offspring simultaneously. What would be the probability in genotype of progeny? Progeny means offspring. Offspring is also called as progeny. If parents were of the genome AB, AB and AB, AB. Given options are homozygous A, homozygous B, homozygous recessive A and recessive B, heterozygous A and homozygous recessive B, heterozygous A and homozygous recessive B. Both are none. The probability of offspring can be easily determined here if we have both kinds of the parents. As you know that these are the linked genes, so this A will combine to this A, and similarly B will combine to this B, B. Cross A and B. So in this case the first option just resembling this, so the option A is correct here. Next question is, if genes are completely or fully linked, it has 100% of gene linkage, then what are the chances of crossing over? Crossing over or recombination? Given options are 0%, 25%, 50% or 100%. As you know that the gene linkage and recombination or crossing over are just opposite to each other. For example, in case of gene linkage, the genes are linked together and are inherited in the same chromosome, while in case of crossing over, the genes shuffle and are not inherited on the same chromosome simultaneously. So both of these are inverse to each other. So in this case, there is 100% of gene linkage, then the chances of crossing over or recombination will be 0%. So the option A is correct, that if there is 100% of full gene linkage, there will be no crossing over. Next question is, crossing over in the non-sister chromatids of chromosomes occur during dash of meiosis. Given options are meiosis 1, meiosis 2, both are none. The non-sister chromatids of chromosome, for example, if we have homologous pair of chromosome, then this chromosome has a chromatid A, then the chromatid B, the other chromosome has chromatid C and the chromatid D. In the chromosome 1, the chromatid A and B are sister chromatids. Similarly, in the chromosome number 2, C and D are the sister chromatids. In case of the chromosome number 1, A is non-sister to the C, and similarly B is non-sister to the D. So if crossing over occur between this B and C chromatids, so this is called the non-sister chromatids. This is normal phenomena, and this crossing over between non-sister chromatids always occur in the meiosis 1, and more specifically in the prophase of meiosis 1. So these were some important kind of MCQs in genetics. Hope you have learned something new in this lecture. Thank you.